Good morning, my dear students of class 8th. Student, today we will talk about the second part of chapter 2nd, the reproduction in plant in biology. And today's topic is artificial vegetative propagation. What is the meaning of artificial? Artificial means a process which is done by man, which is performed by man. This is called artificial, okay, which is against of natural. Several artificial methods have been developed to promote vegetative propagation by horticulturists. Horticulturists are the people who are expert in crop cultivation and management. So by these people, by the horticulturists, few artificial methods have been developed to promote. What is the reason? To promote the vegetative propagation. These methods are also called vegetative propagation and it include cutting, layering, grafting and tissue culture. Let's talk about this process one by one. First of all, the cutting. The cutting method of cutting the stem. In this process, what we do, we just cut a stem into small pieces, okay, and planting the cut end in the soil properly. This is called cutting. What we do in this process is very simple. We just cut the stem and the cut part of the stem is planted in the soil, okay, in the soil. What happens? The piece of stem which is attached to the soil, which is planted in the soil, is eventually bear roots. उसमें जड़ें, पत्तियां ग्रो हो जाती हैं एंड इट बिकम्स एडल्ट प्लांट और बहुत जल्दी वो एक एडल्ट पौधा बन जाता है. यू कैन सी हियर स्टूडेंट, ओके, दिस इज कट पार्ट, okay, of stem and it is planted in the soil. After some time it becomes a new plant, okay. So example of the plant where the artificial vegetative propagation is carried out by the means of cutting are money plant, rose and china rose. So there are few plant, money plant, rose and china rose, etc. These plants grown by cutting method. If you want to grow them, so you should use cutting process, okay. It's very simple process. We just take a stem piece, okay, and that cut part of the stem is planted in the soil. Simply just water it regularly, give nourishment to it, okay, nutrition to it, and after some time it will become new plant after bearing roots and leaves. So it's very simple. Next we have grafting. This method is used to obtain a plant by combining two different plants. In this process we take two different plants. In this process we are combining two different plants, thus it produces a variety. That's why we get a variety with the feature of two different plants. So the new young ones will come after it. It will have the character of both plant, okay. In this method, a stem cutting called scion, okay, scion without roots, scion without root, the part of stem which is without root, upper part is called scion without root. From one plant we take scion of a plant, okay, and then we attach this scion part and tied it to other stem cutting part which have roots, that is called stalk part. So we just attach scion with stalk, okay. This process is similar to cutting. It's same like cutting except the cut part is attached to the stem. Here the cut part is attached to the stem. यहाँ पर हम जो कटा हुआ हिस्सा है उसको stem से जोड़ रहे हैं instead of planting in the soil. Soil में plant करने के बजाय. In the cutting process, what we do, we just plant the cut part in the soil. But here in grafting, we are just attaching that part into the stem of other plant. When a portion of cut stem is grafted on another stem, the cut stem is fixed properly. With the help of tape we do this, so that it becomes a part of host plant, okay. It gets nourishment from host plant and continue to grow. The grafted stem is usually added for more flower and fruits as good qualities of both the plant are combined in the host plant. So the young ones which grown after this process, it has the quality of both plant. Grafting is most commonly seen in rose plant where highly yield of rose flower is achieved, okay. So rose is a plant which can perform grafting as well, okay. So you can see the picture, grafting method used to grow plant. You can see the scion part which is without root and stalk part with root, okay student. So these both parts combined with each other with the help of tape. After some time you can see new branch comes out with it. This is called grafting, okay, and this branch will have character of both plant. So it is advantage of grafting, okay student. Now we will talk about layering. In this method, a portion of the aerial stem is made to grow roots. In this process, what happens, जो वायवीय तना होता है बच्चों, उससे एक ब्रांच निकल के बाहर आती है जिसमें रूट ग्रो होते हैं. कब? इट इज स्टिल अटैच टू द पेरेंट प्लांट. जब वो अपने पेरेंट प्लांट में अटैच होता है, उसके बावजूद, अटैचमेंट के बावजूद वो अब रूट को ग्रो करता है एंड लेटर इज डिटैच फ्रॉम इट. आफ्टर सम टाइम उसको हम ब्रांच को अलग कर देते हैं पेरेंट प्लांट से, जब वो एक इंडिपेंडेंट प्लांट पौधा बन जाता है, ओके. सो in this method, a part of the lower branch of the plant is bent down. You can see here a branch grow from the base of the stem. Okay, it just bent down and touch the soil. You can see here, okay, it touch the soil and layer of bark is removed. And the main thing is that the part of branch which is touch the soil, we just remove the bark from there. Okay, and the part of the lower branch that is touching the soil is then covered, and after that, after some time, we just cover it with the soil and small objects such as small pieces of wood or something else to place it, okay, to place it tightly, and that the branch remains in the soil. Why do we do that? To remain in the soil, that branch part must be in soil. Okay, so to remaining it in the soil, we just put any piece of wood or any something else over it. After a few days, root began to grow from the branch and then the branch is cut off from the parent plant. So जब इसमें रूट पैदा हो जाती है तब हम इसको अपने पेरेंट प्लांट से काट देते हैं. It slowly grow to become an independent plant. So the advantage of this method is that the branch continues to receive nutrition from the parent plant. इसका फायदा ये है कि जो ब्रांच नया पौधा बन रहा है उसको जो नरिशमेंट है, न्यूट्रिशन है, वो पेरेंट प्लांट से ही मिल रहा है. Okay. Layering is carried out in plants such as combining rose, okay, climbing rose, mint and raspberries. So there are a few plants which perform layering, which can be grown with the help of layering process. Okay, climbing rose, mint and raspberries. Now we will talk about tissue culture. Okay, the another process, tissue culture. Tissue culture involves the production of growth of plant in synthetic medium. In this process, what we do, द प्रोडक्शन और ग्रोथ ऑफ प्लांट इन सिंथेटिक मीडियम, एक संश्लेषित माध्यम में हम पौधे को ग्रो करते हैं. कुछ आर्टिफिशियल कंडीशंस होती हैं, कुछ आधुनिक कुछ अवस्थाएं होती हैं, कुछ आर्टिफिशियल है ना, कुछ कृत्रिम अवस्थाएं होती हैं प्रयोगशाला में, जिसके माध्यम से हम पौधे को ग्रो करते हैं. हियर अ काइंड ऑफ प्लांट मे बी ग्रोन इन अ sterile medium. Sterile medium is called the medium where there is no microorganisms, it is completely clean. That is called sterile medium, using a small part of an existing plant in a test tube. So what will you do? We take a few part of a plant, okay, take few part or one part, okay, of a plant, and then we just put it in test tube. Okay, the medium used to grow plant in a test tube. So the medium which is in test tube is rich in nutrients. ये nutrients से rich होता है and suitable for growth of the specific plant. और किसी प्लांट के ग्रो करने के लिए वो माध्यम बहुत सुटेबल होता है. तो ऐसा माध्यम टेस्ट ट्यूब में रखा जाता है. आफ्टर सम टाइम एन अनऑर्गेनाइज्ड मास ऑफ सेल नोन एज कैलस डेवलप फ्रॉम इट. उस समय बाद कुछ उस हिस्से से कैलस बन जाती है, कुछ सेल्स ग्रो करती हैं और कैलस बन जाता है. अब द कैलस इज पुट इन अ हार्मोन कंटेनिंग मीडियम. अब इस कैलस को हम हार्मोन कंटेनिंग मीडियम में रख देते हैं, विच इंक्लूड सेल डिफरेंशिएशन, जिसमें सेल डिफरेंशिएशन शुरू हो जाता है, उसका विभेदन शुरू हो जाता है एंड प्लांटलेट्स का फॉर्मेशन शुरू हो जाता है. द प्लांटलेट डेवलप इनटू एन इंडिपेंडेंट प्लांट व्हेन ट्रांसप्लांटेड टू सॉइल. और ये जो छोटे छोटे प्लांटलेट होते हैं, व्हेन वी प्लेस देम in the soil, after some time it becomes independent plant. Some examples of plant are the cultivated using tissue culture technique are orchid, asparagus and chrysanthemum, etc. So there are few plants which grow with the help of tissue culture. Okay, student? So tissue culture is very simple. What we do, we just take a part of plant, a few part of plant and put it in the test tube. Test tube have medium which is full of nutrients and suitable for growth of plant. After some time callus is developed from it and the callus put it in a hormone containing medium, then it becomes then cell differentiation takes place here and plantlets grow. Okay, and when these plantlets grow into the soil, it becomes new independent plant. So this is how tissue culture take place. Now student, we will talk about advantage and disadvantage of vegetative propagation. Let's talk about advantage. It is faster method. ये बहुत ही तेज माध्यम होता है to produce a new plant. नए पौधों को बनाने के लिए ये बहुत तेज माध्यम होता है. The offspring will be exactly the same as parent. और जो young ones offspring बन के आती है वो अपने parent plant की तरह ही होते हैं. ओके, उनमें वही क्वालिटी उनमें होती है. द प्लांट प्रोड्यूस बाय वेजिटेटिव प्रोपगेशन. वो पौधे जो वेजिटेटिव प्रोपगेशन के द्वारा पैदा हुए हैं, दे बियर फ्लावर एंड फ्रूट्स. उनमें फूल और फल जल्दी आते हैं देन द न्यू प्लांट प्रोड्यूस बाय सीड्स. कंपैरिजन उन पौधों के जो सीड के द्वारा ग्रो होते हैं. तो ये वेजिटेटिव प्रोपगेशन का फायदा है कि इसमें फ्लावर फ्रूट जल्दी आते हैं उन पौधों में दैट इज प्रोड्यूस आफ्टर वेजिटेटिव प्रोपगेशन. इट इज़ अ वेरी यूजफुल मेथड फॉर प्रोडक्शन ऑफ सीडलेस प्लांट. ये उन पौधों के डेवलपमेंट के लिए बहुत अच्छा है जो सीडलेस हैं, लाइक ग्रेप्स और बनाना. उनके लिए वेजिटेटिव प्रोपगेशन इज वेरी गुड. नाउ टॉक अबाउट डिसएडवांटेज. व्हाट इज द डिसएडवांटेज ऑफ दिस प्रोसेस? एज द प्लांट डेवलप्स थ्रू वेजिटेटिव प्रोपगेशन आर आइडेंटिकल. क्योंकि जो पौधे बनते हैं वेजिटेटिव प्रोपगेशन वो सब सेम होते हैं, सेम टाइप के होते हैं, क्योंकि वी आर यूजिंग द वेजिटेटिव पार्ट. देयर इज अ चांस दैट इन केस इफ देयर इज अ डिजीज इन फार्म, तो ये देखा जाता है कि अगर किसी पौधे या फार्म में कोई बीमारी है तो देन इट विल अफेक्ट ऑल प्लांट साइमल्टेनियसली। तो वो बीमारी सभी पौधों को प्रभावित करेगी, एक के बाद एक हर पौधे को प्रभावित करेगी। ये इस प्रोसेस का एक डिसएडवांटेज है। नेक्स्ट, इट कैन रिजल्ट इन द डिस्ट्रक्शन ऑफ एंटायर क्रॉप। अगर ऐसा होता है, if this situation takes place, so what is the result? The result is destruction of entire crop. As there is no dispersal of plant, there are chances of overcrowding. Because there is no dispersal of plants having taking place, that's why there is a big chances of overcrowding. Overcrowding होने के भी chances बढ़ जाते हैं. So this is disadvantage of vegetative propagation, and it was about advantage of vegetative propagation. Okay, so student, it was about artificial vegetative propagation. I hope you must have understood about these all process: cutting, grafting, layering, and tissue culture. All processes are different with each other, okay, and very simple. So it's enough for today. Have a good day, all of you. Thank you.